We have spent the better part of a century thinking about the human body as a relatively self-contained biological system, and we are only now beginning to appreciate how wrong that framing was. The gut microbiome, the approximately 38 trillion microbial organisms living in the human gastrointestinal tract, is not a passenger in your biology. It is, increasingly, looking like one of the drivers. The prediction that routine microbiome profiling will become standard practice at annual checkups is not merely plausible. There is a compelling argument that it is already overdue. The scope of what the microbiome appears to regulate is genuinely startling. This is no longer a story about digestive health. Research has now linked microbiome dysbiosis, disruption in the balance of bacterial communities, to Parkinson's disease, autism spectrum disorder, autoimmune conditions, and treatment-resistant depression. The FDA approval of Vowst, the first microbiome-based drug for recurrent C. difficile infection, was a quiet landmark in medicine. It signals that regulators are willing to treat the microbial ecosystem as a pharmacologically targetable system, not just a biological curiosity. The economic argument for routine profiling is rapidly strengthening. Sequencing costs have followed a trajectory similar to genomic sequencing, collapsing at a pace that makes what was once a research-grade procedure into something approaching a consumer product. As testing costs approach $10 per sample, the traditional justification for not routinely monitoring the microbiome cost effectively disappears. What takes its place is a clinical challenge that the medical community has not yet fully confronted. What do we actually do with this data when we have it at scale? This is where we need to inject some caution. The microbiome research landscape, while genuinely exciting, contains enormous amounts of preliminary and sometimes contradictory evidence. The leap from animal models and small human trials to clinical protocols that treat the microbiome as a primary intervention target is not yet fully justified by the literature. Personalized probiotic prescriptions replacing broad-spectrum antibiotics sounds elegant in theory. In practice, the science of which bacterial strains benefit which patients under which conditions remains at an early stage. There is a real risk that commercial probiotic and microbiome testing industries will outrun the evidence, selling optimization promises to consumers without the clinical rigor to back them up. That pattern is already visible in the direct-to-consumer microbiome testing market today. What the microbiome story does offer, and this is genuinely paradigm-shifting, is a compelling reorientation of how medicine approaches disease. The concept of treating the ecosystem rather than just the pathogen is philosophically profound and clinically important. It moves medicine toward understanding the conditions under which disease arises rather than simply targeting the disease itself. That shift in thinking from disease management to ecological medicine may ultimately prove more valuable than any specific probiotic intervention. The microbiome will become a vital sign. The question is whether medicine will be ready to interpret it responsibly when it does.